అంతే. ఇది మాత్రం ఆపు.
గో లైవ్ అనేది
అసలు ఆపు.
ఓబిఎస్ లో స్టార్ట్ స్క్రీనింగ్, స్టాప్ స్క్రీనింగ్.
రన్ లో ఉంటే స్టాప్ అని ఉంటుంది.
ఓకే.
ఆల్రెడీ స్టాప్ చేస్తుంటే స్టాప్.
ఇప్పుడు మధ్యలో బ్రేక్ వస్తే
దీంట్లోంచి మన ప్రోగ్రామ్ దీంట్లోకి వస్తుంది అనమాట. సో దీన్ని ఇమీడియట్ అనమాట. దీన్ని ఆపేస్తే మన దాంట్లో ఏం జరుగుతుందో అదంతా రాదు. ఇది ఆల్రెడీ అయిపోతుంది కనుక మీరు అట్లా ఏం చేయొద్దు. ఏదన్నా ఉంటే మామూలు ప్రోగ్రామ్ చేసుకోండి. ఏం డౌట్ జస్ట్ కాదు.
కాకపోతే పిల్లలు జాయిన్ అవ్వలేదు ఇంకా. సరే వస్తున్నారు సార్. అదే అన్నాను ముందెళ్ళి అక్కడ కనబడరా ఇటు రావద్దన్నారు. అవుతూ ఉంటాయిలేండి కావాలి. అమ్మో ఇంకా ఆమెకి ఇప్పుడు నేను ఫస్ట్ లెక్చర్ చెప్తున్నాను బండి గుర్తుంటుందేమో. 10-5 minutes గ్యాప్ ఇస్తాను. రండి రాజాన్సి, నేనే పిలిచాను. ఇక్కడ చదవాలి, వెల్కమ్ చెప్పాలి, చదవండి. మీకు ఏమైనా డౌట్స్ ఉన్నాయా ఒకసారి? ఏంటి క్లాస్ ఫస్ట్? పర్లేదు ఒకసారి ఇచ్చేసి వచ్చాను, వెళ్ళండి. Not connected problem. Not connected అని వస్తుంది. అదలా నేనేం చేయలేదు. ఇప్పుడే మీరు చూసారు కదా సార్, రెడ్ది ఓబిఎస్ది. అది నేనేం దాన్ని టచ్ చేయలేదు. అర్థమైంది. టిఫిన్ చేసుకోవచ్చు. రెండు పెట్టి. ఓకే. సార్ చెప్పండి సార్. ఇక్కడ రావట్లేదు అంటే మనకి నెట్ ప్రాబ్లం వస్తుంది సార్, రావట్లేదు. సిస్టంలో ఉన్నా సార్. అదే ఒక్కసారి సార్ రావట్లేదు, నెట్ గూగుల్ ఓపెన్ చేసినా ఓపెన్ అవ్వట్లేదు సార్. చాలా తక్కువే ఉంది కానీ చాలా వద్దు. అయ్యో బాగా మీరు వచ్చారు, వచ్చింది ఇప్పుడు వచ్చింది. ఇదిగోండి ఇప్పుడు గూగుల్ ఓపెన్ అయింది. మరి ఇప్పుడు వాళ్ళు జాయిన్ అయిన వాళ్ళందరూ వెయిటింగ్ లో కనబడట్లేదు సార్. ఉంది ఉంది సార్.
అమౌంట్ మాకు ఆ వేళ 21-22 ఫస్ట్ బిఏ నుంచి 50 మంది నుంచి.
అవును సార్.
ఇది ఎవరు ఫస్ట్ బిఏ అంటే 21లో వాడు ఫస్ట్ బిఏనా అంటే ఇప్పుడు వాడు ఫైనల్ ఇయర్ నాకు వాడు 19లో జాయిన్ అయ్యి 22 ఏంటి నాకు
ఒక్క ప్రోగ్రామ్ అవుతుంది అయిపోయినా మధ్యాహ్నం మీ దగ్గరికి వస్తాను. లైవ్ లో ఉన్నాం, ఆడియోలో ఉంది. ఇదిగోండి సార్ ఇలా వస్తుంది, అవ్వట్లేదు సార్. మధ్యాహ్నం నేనే వస్తాను.
మాకు ఏదో చెప్పిస్తే మధ్యాహ్నానికి వాళ్ళు చెప్తే అది వచ్చి ఫోటో తీసుకొని అందులో ఏది ఎక్కించారో చెప్తే దాన్ని
ఓకే సార్ అర్థమైంది. మీకు 18, 19, 19, 20, 20
మీకు అంతా అయిపోయింది. బీకామ్ ఇది కండక్ట్ చేస్తాం మన
21, 22 దా సార్?
21, 22 ఫస్ట్ ప్రెసెంట్ ఫైనల్ ఇయరా అని అడుగుతుంది.
అర్థమైంది. ఒక్కసారి ప్రెసెంట్ ఫైనల్ ఇయర్స్ మీరు టచ్ అయ్యారు సార్. ప్రెసెంట్ ఫస్ట్ ఇయర్స్ ఎప్పుడు ఫస్ట్ ఇయర్స్
అయితే 21, 22 అంటే
ప్రెసెంట్ ఫస్ట్ ఇయర్
అంతేనా? ఫస్ట్ బిఏ
అదే సార్ ప్రెసెంట్ ఫస్ట్ ఇయర్స్
అంటే సెమ్ 1 వాళ్ళేనా?
అంతే. సెమ్ 2 అదే సెమ్ 1 థ్యాంక్ యూ సార్. మాకు గుర్తుంది లేండి సార్, ఇస్తానండి. మాకు గుర్తుంది. అంటే ఇక్కడైతే కనెక్టెడ్ అనే కనిపిస్తుంది. ఓకే కానీ ఇది కొంచెం గూగుల్ కూడా అదే వాళ్ళు జాయిన్ అవుతుంటే అక్కడ ఇప్పుడు గొప్ప కష్టమే.
ఆవిడ అలగడం
రండి రా.
అదేదో 6 రూపీస్ రావాలి
అది కూడా
సేల్ ఉందా అతనికి సార్ చెప్పేసి సరే నేను మధ్యాహ్నం వస్తాను
సార్ ఇంకొక అమ్మాయి ఇవ్వలేదు ఎగ్జాంపుల్
ఎవరు
తులసి అని
ఏమమ్మా
రాలేదు
ఒకసారి ఫోన్ చేశాను
సార్ ఆమె దగ్గర ఫోన్ లేదు నేను ఒకసారి క్లాస్కి వెళ్ళి
సరే
యా ఎవరి మీద నేను నువ్వు మాట్లాడేదా ఏమన్నా కొంచెం వాటర్ తాగుతా ఏమో వాటర్
చెప్పండి
సరే
ఎస్ మేడం టుమారో షూర్ మేడం
ఇది ఫస్ట్
ఎస్ మేడం
ఇది చదువుతాను
టుమారో విల్ అటెండ్
ఆ తర్వాత సాంగ్ ఇంకా డౌన్లోడ్ ప్లే చేస్తాను సాంగ్ అయిపోయినాక ఓపెనింగ్ రిమార్క్స్లో ప్రోగ్రామ్ కన్వీనర్ డాక్టర్ సి శాంతి తర్వాత ఐపీఎస్సీ మేడం అయిపోయినాక థ్యాంక్ యూ మ్యామ్ అని చెప్తాను తర్వాత రిమార్క్స్లో ఐపీఎస్సీ కోఆర్డినేటర్ డాక్టర్ థ్యాంక్ యూ సార్ ఫర్ యూర్ ఎంకరేజింగ్ వర్డ్స్ అడ్రస్ బై చీఫ్ గెస్ట్ డాక్టర్ కె శ్రీకాకుళం థ్యాంక్ యూ వెరీ మచ్ మేడం ఫర్ యూర్ ఇన్స్పైరింగ్ వర్డ్స్ అని చెప్పి చిన్న షెడ్యూల్లో మార్పుందా నేను చెప్తానండి యాజ్ ఆఫ్ నౌ ఒకసారి చదువుతాను ఏమండి
సరే సరే పిల్లలందరూ జాయిన్ అవ్వండి చెప్పాను మేడం అందరూ అవుతున్నాను కొంచెం నెట్ స్లో ఏదైనా మనం చెప్పాము మనం చెప్పిన మెంబర్ ఒకటైతే మనం చెప్పలేదు ఇలా నన్నే అడిగింది అన్నమాట మేడం యాక్చువల్ నాకు లీవ్లో చేసేది కాకుండా ఇది ఒక దరిద్రం, చేసేది కాకుండా ఇది ఒక దరిద్రం. ఈ ఎంతలే మేడం, ఎప్పుడంతే. అంతే అంతే మేడం, ఆ మాటలు మీరు పట్టించుకోవాలి, అవి మనం చేసుకుని వెళ్ళదు. అంతే కదా, మనం ఊరికే పెట్టేస్తున్నాం మనమేమో. అంతే కదా, అవును. ఆవిడ ఏంటంటే అదేదో అదొక రకం మేడం, మనం అదే కదా. అంటే ఈ ప్రోగ్రామ్ చేయడం అంత ఈజీ అనమాట. కోఆర్డినేషన్ చేసుకొని చేయాల్సిన ప్రోగ్రామ్. అదే అది నేను చెప్పాను. నేను యాక్చువల్లీ మేడం కూడా రాలేకుండా హెల్త్ సరిగ్గా లేదని అన్నారంటే అది మీరు అన్నట్టే. మేడం ఇది ఇందాక మనం పెట్టిన లింక్ అయిపోయింది మేడం, ఎక్స్పైర్ అయింది ఏంటిది? ఒకసారి సార్ జాయిన్
సార్ ఒక్కసారి పిల్లలకి పెట్టిరా ఒకసారి. అది ప్రింట్ తీయలేదు బ్రో సార్, సాఫ్ట్ కాపీ ఉంది. అది ఒకసారి చూసి పాపులర్ లెక్చర్ సిరీస్, అందులో టైటిల్ ఉండదు కానీ సార్. నమస్తే సార్, కూర్చోండి. ఒక్కసారి సార్, ఒక్క మీకేదో జాబ్ వచ్చిందని విన్నాను, కరెక్టేనా? మీ గురించి అన్నీ వస్తున్నాయి, జాబ్ వచ్చిందని, ఏదో ట్రైనింగ్కి వెళ్తున్నారని. ఫీజు సంగతి ఓకే, మధ్యాహ్నాల క్లాసులు ఉండరు. ఈరోజు నిన్న రేపు కాదు, ఇన్ని రోజులు ఎందుకు లేరు చెప్తే. చెప్పండి, మీనట్టే ఇది కూడా అవుతుంది, అవ్వట్లేదు అసలు. సార్ లింక్లో మీరు జాయిన్ అయ్యారా? ఒక్కసారి చూడండి సార్, అవ్వట్లేదు ఎందుకో లింక్. ఒక్కసారి గ్రూప్లో పెట్టారు సార్. అక్కర్లేదు సార్, రిజిస్టర్ గూగుల్ మీట్ లింక్ ఒక్కసారి. ఏంటి ఇప్పుడు ప్రోగ్రామ్ అవుతుంటే 5 వరకు ఉంది కదా, 4:30కి వచ్చినాను కనబడండి. వెళ్ళండి. ఎక్కడికి వెళ్తారు ఆయన ఎక్కడికి వెళ్ళారు? 10 to 5 కాలేజ్ అంటే ఎవరు ఎక్కడికి వెళ్ళిపోవడానికి ఉండదు.
తర్వాత someone lets me in
అవును, వెయిట్ అంటిల్ సమ్మన్ లెట్స్ విన్ వస్తుంది కానీ ఇక్కడ అవ్వట్లేదు కదా. కానీ ఇక్కడ నాకు కనబడట్లేదు సార్. టు లెట్ యూ ఇన్ అనేది నాకు ఇక్కడ కనబడట్లేదు. సార్కి ఇది ఇచ్చినట్లేదు నేను. అక్కడ కట్టట్లేదు సార్. టు లెట్ యూ ఇన్ అనేది నాకు ఇక్కడ కనబడట్లేదు. సార్కి ఇది ఇచ్చినట్లేదు నేను. అక్కడ కట్టట్లేదు సార్. టు లెట్ యూ ఇన్ అనేది నాకు ఇక్కడ కనబడట్లేదు. పట్టలేదు సార్. పట్టలేదు సార్.
అన్ని కాదు ఎగ్జాంపుల్ ఇచ్చిన డబ్బులు ఎంత ఇవ్వాలండి ఎగ్జాంపుల్ అది అసలు మొత్తం కూడా అవుతుంది మూడు అలా మార్నింగ్ వచ్చేసింది డెలివరీ బాయ్ వస్తే స్టార్ట్
చిన్న పాప ఎంత ఏజ్ సార్? ఇంకో లింక్ క్రియేట్ పిల్లలకి అలాగే వస్తుంది సార్.
ఇంకొక లింక్ క్రియేట్ చేసి నా దాంట్లో చేసి
అవ్వట్లేదు.
ఇంకొకటి చేయండి.
వస్తుంది. ఇంకో లింక్ క్రియేట్ చేద్దాం అంటే
మేడం
ఫ్రెష్ లింక్ క్రియేట్ చేద్దాం అలా కనిపిస్తుంది సార్. ఇప్పుడు చూడండి ఇది అవ్వట్లేదు. ఇంకో లింక్ క్రియేట్ పిల్లలకి అలాగే వస్తుంది సార్.
ఇంకొక లింక్ క్రియేట్ చేసి నా దాంట్లో చేసి
అవ్వట్లేదు.
ఇంకొక దీంట్లో వస్తుంది మేడం, యూట్యూబ్లో వస్తుంది. టైం ఎంత అయింది సార్? 11. ఏమన్నారు సార్ మేడం? ఐ డోంట్ హ్యావ్ టైం. నాకు జాయిన్ అయింది, జాయిన్ ఇట్లా వచ్చింది. సరే ఇప్పుడు మీరు పంపింది మేడం.
10 to 5 కొత్తది.
సేవ్ సేవ్ అయినట్టు.
పెట్టింది ఎవరు?
మేడమే పెట్టి లీడర్లు ఏం చేస్తున్నారని అడిగారు.
అది గ్రూప్ లో పెట్టేసారు.
జాయిన్ అవ్వండి.
కొత్త లింక్ పెట్టారు. శాంతి కొత్త లింక్ గ్రూప్ లో పెట్టారు. పిల్లలందరికీ మీకు పెట్టాను.
ఇందాక యాక్చువల్గా
ఒకసారి అందరినీ
కాల్ చేసేది.
నేను పెట్టింది నువ్వు పెడతాను గ్రూపుల్లో. నేను అన్ని మా కాలేజ్ గ్రూప్లో పెట్టను. అది కొట్టి కొట్టి కొట్టి
నేను ఎటు వచ్చాను. మీరు స్పీకర్ అయినా పెడతా మేడం. ఒకసారి నెట్ కనెక్షన్ పోయిందండి. దీంట్లో ఓపెన్ చేసి ఇది ఉంది కదా, దీన్ని ఓపెన్ చేసి డిస్కనెక్ట్ చేసి మళ్ళీ కనెక్ట్ చేయండి ఆటోమేటిక్గా. మంచి వాళ్ళు.
Good morning, dear participants. I'm Shanti from DBT Technology are sponsoring the seminars, symposium and workshops to conduct and to popularize the every students and every lecturers and every participants and encouraging them. And this program mainly based on the environmental sustainability. To protect our environmental, we are giving this lecture. So I hand over this to proceedings to IQAC coordinator Dr. Poli Naidu sir.
Dr. Sureka Madam Garu and coordinator of this program Dr. Santi Devi Madam and so many other participants from different colleges of the state. Madam Santi Devi Garu, who is very keen in developing some research based techniques and programs like this for the benefit of the students. I congratulate Dr. Santi Devi Garu and all the best to you, madam. Thank you for this wonderful opportunity.
Thank you, sir, for your kind and encouraging words. So, dear delegates, just give us 5 minutes and we'll start with the schedule. మనకుదేలే కదా సార్, ఇట్స్ ఓకే. ఆ సార్ రాలేదు, నేను చెప్తున్నానని చెప్పేసి ఫోటో తీసాను. ఏంటో అవ్వట్లేదు. Yeah, right. So I think we are live now. Not a problem. We had some technical issues, so thank you. Technical issues, so thank you for your patience. And today I'll be talking about the biotech. I was saying climate change is one of the greatest challenges facing this generation. The rapid accumulation of anthropogenic carbon dioxide in the atmosphere is already altering the natural climate and biological systems, which in turn is resulting in abnormally destructive wildfires, storms, rain patterns, and the spread of infectious diseases. It is increasingly clear that the historical fossil fuel-based models of carbon, energy, and material cycling through the economy are incompatible while maintaining a hospitable environment. So humanity will need to bring every tool it has to bear on this critical challenge. New approaches are required at almost every field of the economy, and biotechnology definitely has the potential to be a transformative asset in this struggle. The reason being that biotechnology applications touch most aspects of modern life, they being plants and other living material. But in the past 150 years, much of our economy has come to depend upon petroleum and other non-renewable resources. And the environmental consequences of this transition from renewable sources to non-renewable resources are well documented. Biotechnology has developed more sustainable, bio-based alternatives for many products, including fuels, polymers, and other chemicals. But ultimately, they still require a material input, reduced carbon footprints and environmental benefits compared to the traditional systems that they are displacing. And these are reliable and cost effective. These alternatives depend on broad availability of sustainable biomass feedstock. At present, there are concerns that there is not enough biomass to sustain the available meat to growing demand. So biotechnology is rapidly reducing the carbon footprint of feedstock production by enabling new sustainable ways to produce usable biomass, improving yields on existing crops, methane that is being used today, simply by switching the source of the gas. The manufacturing sector is responsible for 22% of the total greenhouse gas emissions. And while no single technology or solution can single-handedly solve this problem, biotechnology enables opportunities for lower emission production across many sectors. The developing of lower carbon products. In this, as awareness of this climate crisis expands, consumers are increasingly demanding low carbon options and more sustainable replacements for existing products. This means finding low emission alternatives that provide the same level of performance, durability, and cost effectiveness as mature fossil-based systems. Here, biotechnology allows for the production of low-carbon consumer products through the substitution of biomass or other recycled carbon feedstocks, and by enabling more efficient, biologically based production. About what a sustainable future may look like, several features are common across all scenarios. One of these is the deployment of massive amounts of carbon capture and sequestration, which is CCS, carbon capture and sequestration, which converts carbon to a form that does not contribute to climate change, or it stores it underground. CCS cannot be the sole or even the primary solution to the climate change, but take into consideration that it will make a carbon products and enhance our carbon sequestration. So in the coming slides, we'll be looking at climate mitigation, and which can be used for as a solution which provide a biotechnological solution to solve these environmental problems that we have brought upon ourselves. So coming to these are the four broad categories under which we'll be looking at the different biotechnological products. So one is the products itself, and the other one are the agricultural inputs and the climate services. And the next one is the biotechnological tools and bioindustrial manufacturing. So what are the advances that are going on in your industrial manufacturing units and plant and biotechnology technologies. So coming to the products. Within the products, we'll study it under different heads. The first one being advanced biofuels. So what actually are biofuels? So liquid biofuels were one of the earliest biotechnology products to be deployed at a large scale in the world for the purpose of achieving this greenhouse gas emission reductions. In the early 21st century, production mostly took the form of first generation biofuels, that is the ethanol and biodiesel, bioethanol and biodiesel, that were derived from feedstocks such as corn and vegetable oils. But the problem that arose here was that these feedstocks are also of starch-rich feedstocks to ethanol. and to a lesser but still substantial extent the trans-esterification or hydro-treating of vegetable oils to biodiesel or renewable diesel as it is called. So a shift to more sustainable growing practices that has been driven by a desire to capture the compliance values of low carbon programs is further reducing the carbon intensity of the first generation fuels. So recent progress in the development of biocatalysts and engineered microorganisms has made it possible for the production of ethanol from these second generation feedstocks such as grasses, shrubs and other dedicated energy crops. So here you can see how the shift has happened. In the first generation, biofuels were produced from important feedstock biomass like corn and vegetable oils, while the second generation we are aiming towards these grasses and shrubs which are not the feed employs biocatalysts that tend to break down the cellulose and the hemicellulose, which are the major components of grasses, to glucose and other constituent sugars. The glucose is then converted to fuel ethanol in the same manner as in corn glucose is being converted. So microorganisms that are naturally able to ferment glucose have been engineered using biotechnology to make them more capable of also fermenting the simple sugars that are in turn derived from hemicellulose to ethanol. So initially these microorganisms tend to use starch when we have engineered them such that they can also ferment the simple sugars that come from the digestion of your hemicellulose to ethanol. So what we are doing, we are improving both the yield and the efficiency of lignocellulosic biofuel production. An early commercial application of this pathway utilizes this lignocellulose, that is lignocellulose, that is found in small quantities in corn kernels to produce ethanol. So corn kernels are the stalk or the base that we throw off after you eat the corn. So these corn kernels are used as a source of ethanol. Biotech companies like POET, Syngenta and Inogen, among several others, have begun adding corn kernel fiber conversion units to this first generation broad deployment of this cellulosic technology to agricultural residues, municipal solid waste and dedicated energy crops. So this has to have a wide application. Advances in biotechnology have expanded the major supply of feedstocks that are available to biodiesel and renewable diesel. Two of the major success stories are in sustainable transportation. So biodiesel, BD, which lipid feedstocks are reacted with methanol to yield a fatty acid methyl ester, FAME. This in turn can be blended with the conventional diesel without needing any further modification to the engine. So you have to appreciate the fact that we are using biodiesel along with the original common conventional diesel in the normal engines. You do not need to make any kind of modifications to the engines and still these can be used. And you have the renewable diesel, that is the RD. So small difference, you have a biodiesel and you have a renewable diesel. Both are the products of biotechnology. So renewable diesel is made from hydro-treating the same kind of lipid feedstocks in a process that is very similar to parts of oil refining. But it has the performance characteristics like those of a diesel fuel. So this renewable diesel, which is derived from the lipid feedstock, can also act like a diesel, conventional diesel fluid. So biobutanol has more chemical characteristics and it more strongly acts like a hydrocarbon than the original ethanol does. So this biobutanol is produced via fermentation from the same simple sugars as in ethanol production. Some biofuel producers have the production of ethanol has been genetically modified so that it can produce isobutanol. And there are also pathways that utilize bacteria. Instead of yeast, we also have bacteria for used in the production of your isobutanol or biobutanol. So biobutanol can also be produced via engineered microorganisms from the carbohydrates in some microalgae strains. So you have microalgae that can produce biobutanol, you have yeast that can produce, and you have even a bacteria that can produce bioethanol. But remember, all these are genetically engineered microorganisms. So this is where the application of biotechnology comes. Butanol has in fact attracted attention or interest as a key step during for production of renewable hydrocarbon fuels, isooctane, that is fertilizers, pharmaceuticals, additives, explosives and adhesives. We mostly read that petroleum chemicals are used in the production of petrol and other fuels, but you have several other common products that we use in our daily life that are being produced or derived from these petrochemicals. So all these differ from the fossil fuels in that their consumption does not normally cause these greenhouse gases emission via combustion. This might be the reason why while we always read under the pollution heading, we just tend to read directly the petroleum products, the direct petroleum products have been developed by biotechnology industries. So the ability of biomass to replace a wide variety of fossil products has greatly benefited from recent biotechnological advances that have enabled the manufacture of products from both the categories. So we are either completely replacing the use of these fossil products by these biomass derived products, or normally it is produced from a natural gas or petroleum feedstocks. So what we are doing is these building blocks are obtained, we are obtaining them either from ethanol, as in like your bioplastic ethanol, or from hydrocarbons, both being converted from your biomass feedstocks. So what we are doing, the starting material, that is the fossil feedstock, is being replaced by the biomass feedstocks. And we had already actually discussed this. So growing concerns over the longevity of plastic waste in the environment has also prompted the development of biodegradable plastics that are capable of decomposing over short time frames when compared to your traditional plastics. So the most common of these examples are polylactic acid PLA and polyhydroxyalkanoates PHA. So how do we derive this? This PLA, that is polylactic acid, is derived from plant sugars which are naturally fermented by bacteria to yield lactic acid. This lactic acid is then chemically converted to PLA, that is polylactic acid, which can be used as a bio-based plastic. So this is how this biodegradable plastic is being synthesized. And you have this PHA, polyhydroxyalkanate. Even this is produced via fermentation of plant sugars. So the starting materials, always the same point, they are biomass. So biotechnology breakthroughs have also been made in the lesser known but equally important fossil product, that is the lubricants. So we are aware, everybody is aware, petroleum products, yeah, it is the biofuels and the plastics, the most commonly known petroleum products. But another petroleum product is also there which plays a very important role in this GHG emission, that is your lubricants. So lubricants are actually made from petroleum and are a common use throughout your industrial and transportation sectors. While they actually represent a small share of your refinery product mix, but they are critical. Though they are used in small quantities, their role is very vital. So what we are doing, what biotechnologists are doing, are that plant sugars, they are exploring the fact that plant sugars can be fermented by bacteria to yield a chemical that is capable of conversion to bio-based versions than their petrochemical counterparts. So these bio-based products have less energy input. They have less energy requirement when compared to your petrochemical products. And secondly, these bio-based products are built from carbon that would otherwise reside in the atmosphere and thus serve as a vital pathway for atmospheric carbon removal. So what are the starting products that we are here using? We are using the biomass. If we will not use the biomass, it would stay in the environment and contribute and increase the carbon content of your environment. So in one turn, what we are doing, less energy input requirement pathways, and in the other side of the coin, we are removing the biomass, that is, we are removing excess carbon from the environment. Thus, biotechnology provides a wide range of options for reducing the carbon intensities of many bio-based chemicals and products. Now, let's see what these food and feed ingredients are. So, in food and feed ingredients, we have plant-based products, proteins and food products. So, the global food system, including the land and the resources that are required to raise animals, grow crops, process, package and transportation. So all these, that is the global food system as we call it, is responsible for dairy options. So in future you will be having meat and dairy that have been derived from plant products. So one of the more novel applications of biotechnology is cultured meat products. New Age Meats is one of the several companies that is working to produce cultured meat. So what is this cultured meat? It is an engineered tissue that is produced in laboratories by microorganisms that induce and feed the growth of animal muscle cell in a bioreactor. So we are producing meat in a bioreactor using microorganisms from plant-based products. Look at the paradox, right? So this cultured meat production is an energy intensive process that is there. So because it is such a complex product and it involves so many factors and variables in it, this is definitely energy intensive. So energy input is more when compared to your simple poultry production. But this meets lack of methane production and ability to utilize low carbon energy sources is projected to reduce our GHG emissions up to 96% when compared to traditional meat products. So imagine traditional or other ingredients of animal feed. So we are taking these greenhouse gases and trying to convert them into animal feed. So in addition to recycling these greenhouse gases that would otherwise be emitted directly into the atmosphere, which is known as gas fermentation, our biotechnological methods are trying to reduce the use of the land, avoiding the largest source of GHG emissions associated with feed products. So like we have mentioned in the starting, this feed and food ingredients, the GHG emissions from the food and feed emissions are being tackled at different angles. For all animal agriculture emissions is attributed to methane from enteric fermentation in the digestive system of these ruminant animals. So in the digestive system of the animals itself, you have around 40% of the methane emission, and our biotechnology is trying to target that 40%. So biotechnology solutions are being developed to address emissions from cattle, swine, poultry and other animals. You have biotech enzymes from Novozymes and others underway to utilize biotechnology to improve the efficiency of anaerobic digestion through the optimization of microbes and microbial communities used. So you know the digestion, yeah, we have microorganisms, even we, all living organisms have microorganisms as symbiotes in their digestive system which help and aid in the digestion process. So we are also trying to target those microorganisms and reduce the carbon output. Coming to food and feed waste. So earlier we had seen food and feed ingredients, and now we will see how food and feed waste are how we are trying to reduce the GHG emission from food and feed waste. Waste from food and feed production and delivery is also a significant source of the GHG emission. Nearly a third of all the food produced is waste. So ultimately what we are doing is we are trying to decrease the food spoilage so that the waste in turn can also be decreased, and the carbon footprinting, the waste of the carbon waste that. So that was all the again studied under two heads. You have your agricultural biologicals and you have your biological carbon capture, use and storage. So what is this agricultural biological? Modern agriculture, as we all know, is an energy intensive process. In addition to the need of fuel, heavy metals. So here see that all our agricultural practices are in fact increasing your carbon dioxide concentration indirectly, but still. Nitrogen fertilizers increase the sequestration potential and minimize the land footprints of crops. So nitrogen fertilizers do have the advantage in that they help in sequestering of this carbon and minimizing your carbon footprints. But the environment. So biomass is one of the world's major carbon sinks. So the concentration of the carbon in the world is in your biomass. All forms of biomass that employ photosynthesis will capture atmospheric carbon dioxide and convert it to carbon-based compounds like sugars, starch, and lignocellulose. The carbon content of this biomass remains sequestered until the biomass is either consumed or decomposes. So until it is within the plant, not a problem for us. But the moment it is consumed, the plant or the plant products are consumed and the carbon dioxide from the gases or to remove carbon prior to combustion itself. CCS can also be deployed at facilities utilizing biomass as a feedstock. The biomass energy capture and sequestration. BECCS. CCS is just carbon capture and sequestration, whereas your BECCS is biomass energy carbon capture and sequestration. So these processes actually reverse the past emissions. So look at this. We are not just working on not releasing new carbon dioxide into the atmosphere, but we are trying to reverse our emissions, past emissions. That which has already been released into the environment can also be sequestered using this technology. So the biomass captures atmospheric carbon dioxide during its growth phase. It is then combusted, which will yield both energy and carbon dioxide. The bioenergy displaces the fossil energy, and the carbon dioxide that is released is either sequestered in underground caverns as a gas, or it is converted to a degradation resistant solid such as a biochar. So we are not releasing the carbon dioxide into the environment. You either store it underground in caves as a gas, you seal the gas, or as a non-degradable solid which is known as a biochar. So BCCS, Biomass Energy Capture and Sequestration System, is a carbon negative process. How? It results in more carbon dioxide being sequestered than emitted. Technology advances that increase the growth rate, growth potential, and harvest efficiency of the biomass which is used in your BECCS stock enhances the process's carbon sequestration capability. So I hope you can appreciate the fact that we are even trying to retrace our carbon footprint. Environment due to our human activities can also be retraced and recaptured back using biotechnology tools. So now moving on to the new biotechnology tools and bioindustrial manufacturing that can be used for your climate mitigation. So in the new biotechnology tools, we have rapid advances, typically increase the speed and reduce the cost of genetic engineering global challenges, including climate change. Coming to applications of biomanufacturing in traditional industries, some of the industrial biotechnological earliest uses were in the applications of enzymes to improve the efficiency and reduce energy use in traditional industries. The introduction of enzymes for pulp and paper bleaching, for example, reduced energy consumption around a number of companies such as you have your Ginkgo Bioworks, Aritzia, Twist Biogenes are providing organisms design and DNAs. So why do we need to synthesize or manipulate your microorganisms? to optimize the manufacturing pathways in which these microorganisms are utilized. So a shift to biological data storage eliminates the need for mining and production of silicon information in DNA. Instead of using your silicon chips for data storage, scientists are actually working on constructing DNA. We all know that DNA is a genetic information carrier. It carries the genetic information. So what we are working on is constructing synthetic DNA which will carry our data. So energy consumption by data storage facilities already accounts to 2% of your global GHG emission. And in fact, this is projected to surge or increase to 14% by 2040. So these biology-based parallel computing, in which your biomolecules are used for the energy, carbon removal, even small improvements in this application, that is the plant and animal biotechnology. So as we have seen, and I have been repeating from the starting of the lecture, so these past breakthroughs have mitigated climate change by reducing the amount of land required for agricultural sector. It is important to note that the GMO's productivity increases during extreme weather events will improve both climate and energy security. I do still agree that agriculture is important, but what we are doing is we are destroying the natural ecosystems, the forests and your grasslands and other ecosystems, and we are converting them into agricultural land. So agricultural practices, this accounts for even your civilization activity. We are doing deforestation of the forests, the grasslands, the open lands that are there, and we are destroying all the greenery in it. So in turn, again, we are opening our carbon sinks. So biotechnology is enabling the expansion of these existing bioenergy pathways. Work is underway to utilize fast carbon sinks. However, their utilization for bioenergy has historically been constrained by poor crop yields outside of laboratory. So though they have the advantage of marginal land growing, but the disadvantage is that their efficiency has been obtained only in a laboratory, not outside it. So GMO has slowed the progress. Finally, our CRISPR gene technique has also been employed in improving the resiliency and carbon efficiency of your first generation bioenergy feedstocks. The one we had talked about in the first, expected to occur with growing frequency as a result of climate change. So what we are doing, we are trying to increase the resiliency of these feedstock, bioenergy feedstock crops to extreme weather conditions. So biotechnology is also used to produce plant varieties including apples and potatoes. This we have already seen having extended shelf life to avoid cosmetic spotting that causes consumers to throw away the food and again increase the waste and in turn lead to carbon increase. Biotechnology has enabled major improvements in the yields, land use efficiency and resiliency of important bioenergy feedstocks in the recent decades. Moving on to animal biotech. Until now, we have just talked about all the solutions and the tools of biotechnology that are being utilized. But you have to remember that there are several hurdles that are being faced by the biotech industries at every stage, which include you have ethical barriers, you have your financial barriers, policy barriers, regulation barriers. These are just one among the many. So yes, we have come a long way and we still do have a much longer way to go forward to. But climate change, as we have said in this slide, will affect every person, every nation, industry and culture on earth. There is no one who can say that we will not be affected by the climate change. And to conclude, biotechnology offers vital contributions to both the near-term GHG reductions and revolutionary tools to combat climate change in the longer term. You have both the short-term solutions and the long-term solutions that are being offered by biotechnology. To successfully address the challenge of climate change, rise the full potential of biotechnology to combat climate change. The further growth of the world biotechnology industry will be heavily affected by both the existing and the potential barriers and by the degree to which the governments invest in the development and deployment of biotech solutions. Biotechnology is a vital component of the national and global infrastructure needed to combat the catastrophic climate change. The economy-wide scope of this challenge will require the adoption of policies that reflect the ability of biotechnology products to achieve decarbonization across all major sectors of the world economy. Biotech companies will need to speak up not only to ensure the new policy provides opportunities for success, but to also make it clear that prosperity is not threatened by sustainability. We can have both. There is ample evidence that reducing emissions, in fact, is essential in supporting a thriving economy. Biotech industry has a tremendous opportunity to build upon decades of success and provide critical tools and expertise for decades to come. Like every other industry, change will be profound and lasting. But if any industry can demonstrate that change can be an opportunity for growth, this is the one. With this, I would like to end my talk, and I would like to acknowledge the DBT New Delhi for sponsoring this popular series lecture, and also I would like to express my gratitude to my graduate final year students Rajesh and Pavan, who have put up done all the background research for the talk that I have just given. Thank you, Rajesh and Pavan, and I also thank the organizers for giving me this opportunity. I would like to take the questions, if any, now. So do we have any questions? Right, so in the absence of questions, what we'll do is we'll take a break of 15 minutes and then we'll reconvene for the next lecture. Thank you, and see you soon. Thank you. పిల్లలు 21 మంది ఎందుకున్నారు? 21 మంది ఎందుకున్నారు? ఝాన్సీ, నిఖిల్ డౌన్లోడ్ చేసి ప్రదీప్ పెట్టావా? యా, అమ్మా మజ్జిగ తాగేవా? ఒక్క నిమిషం ఇది కూడా ఈరోజు పడుతుంది మరి. ఒకసారి ఆఫీస్లో వచ్చి కట్టుకోండి. వేసాం మరి ఎంటికల్ అంటే అది నెక్స్ట్ డే, మరి ఇంకా ఇప్పుడు థర్డ్ డే అవుతుంది. అన్నీ అయిపోయి అంటే ఒకసారి రండి, ప్రిన్సిపల్ మేడంకి చెప్తే ఒక్కటే ఉంది అంటే ఒకసారి ఇన్స్టెంట్ ఎగ్జామ్ ఏమైనా పెడదామని యూనివర్సిటీకి పెడదాం. ఒకసారి ఎలాగున్న మీ వాళ్ళు ఎంతమంది ఉన్నారో ఇలాగా వాళ్ళందరూ వస్తే ఒక సబ్జెక్టే ఉండిపోయిన వాళ్ళు ఒకసారి రండి అండి. ఇన్స్టెంట్ అంటే ఒక ఎగ్జామ్ ఉంటేనే కండక్ట్ చేస్తాం. ఓకేనా? ఆ పని చేయండి. ఎంతమంది ఉన్నారో కనుక్కొని అందరూ మా సార్ దగ్గరికి వస్తే ఆ పని చేద్దాం. సరే, ఓకే. ఏరి వీళ్ళందరూ జాయిన్ అవ్వలేదు? అందరికీ నెట్ లేదమ్మా. ఇక్కడ మీ పేర్లు చూపిస్తాను ఎంతమంది జాయిన్ అయ్యారు. ఏంటి కాదు, ఇప్పుడు మన ఈవెంట్కి మనమే జాయిన్ అవ్వకపోతే ఎలాగ? 17 మంది ఉన్నారు. ఆ 17లో మేడం ఒకరు, నేనొకరు, సార్ ఒకరు. ఏంటిది మొత్తం? చెప్పానా రిపోర్ట్? ఆ రిపోర్ట్ ఎంత? 40 పేజీలు తీసి ఇప్పుడు జాయిన్ అవుతున్నారు కదా. అంటే నువ్వు వస్తే జాయిన్ అవుతారా? ఉండు మా వాళ్ళకి చెప్తా. నేనేమన్నానంటే మా వాళ్ళకి కెమిస్ట్రీ అంటే చాలా 3 ఇయర్ ఫస్ట్ ఫైనల్ ఇయర్కి కెమిస్ట్రీ 3 గంటలు. అదే జువాలజీ వాళ్ళు వీళ్ళకేమో జువాలజీ. నేనేమన్నానంటే జాయిన్ అవ్వండి, సైలెంట్లో పెట్టుకోండి. అంటే ఒక్కరు జాయిన్ అవ్వలేదు. ఉన్నారులే ఒకలో ఇద్దరు, నాకు పేర్లు కనబడదు. అదే వాళ్ళు ఫోన్ చేద్దామని ఫోన్ తీసి కానీ అలా కాదు. నేను ఇందాక ఫీల్ అయ్యానమ్మా. నేనేవో నీకు పెద్ద దీనిలా నేను చూసుకుంటాను, నువ్వు పర్వాలేదులే అన్నాను అంటే అలా కాదు. తప్పు ఎక్కడ జరిగింది అంటే అది ఒక్కటేనా, ఇంకేమైనా తప్పు? ఆవిడ చెప్పలేదన్నా. జ్యోతి జాయిన్ అవ్వమనండ్రా, అందరు నిన్ను చంపి పడేస్తాను. సైలెంట్లో పెట్టమనండి. మనం గ్యాంగ్ అంతా ఇక్కడే నించుంది బయటే, నేను చూసాను. జాయిన్ అయితే ఏమైపోయింది? అవును, జాయిన్ అయ్యి సైలెంట్లో పెట్టుకుంటారా? అంటే ఇక్కడ నెంబర్ తక్కువ ఉంది మా, ఎస్పెషలీ మధ్యాహ్నం ఉండండి. మీకు నెట్టే ప్రాబ్లం అంటే మధ్యాహ్నం ఇంపార్టెంట్. 1:45కి ఎక్స్టర్నల్ వస్తున్నాం, అప్పుడు కొంచెం అందరూ జాయిన్ అవ్వండి. ఆ సెషన్ మాత్రం ఫుల్ ఉండాలి. ఓకేనా? సరే, కొంచెం మీ సెకండ్ ఇయర్స్కి ఫస్ట్ ఇయర్స్కి కూడా చెప్పండి. ఆఫ్టర్నూన్ సెషన్ కంపల్సరీగా ఫుల్ ఉండాలి. ఇప్పుడు పర్వాలేదు. సరే, ఓకే. కెమిస్ట్రీలో ఉన్నామండి. జ్యోతి జాయిన్ అయింది, మధ్యలో పోయింది. జాయిన్ అవ్వండి అన్నారు, మళ్ళీ నేనే అన్నాను వద్దు మధ్యాహ్నం ఉండాలి, నెట్ పోయింది అనుకో, వీళ్ళు నెట్ అయిపోయిందని చెప్తాను. నేను అన్నాను సరే 1:45కి నాకు అందరూ జాయిన్ అవ్వాలి, 3 ఇయర్స్ మొత్తం నాకు కనబడాలి, ఎక్స్టర్నల్ స్పీకర్. ఆ టైంకి మాత్రం అందరూ ఉంటారు. అందుకే వద్దాను. దాని పేరు తన ఫోన్ ఇప్పుడు వద్దులే. అప్పుడు 1:45కి షార్ప్ జాయిన్ అవ్వమని సార్. సందీప్త జాయిన్ అవుద్ది కదా. సందీప్తకి కొత్త లింక్ పంపించు. పెట్టావు కదా, ఆవిడకి మళ్ళీ చేద్దాంలే. 1:30కి ఫోన్ చేద్దాం. ఉన్నవాళ్ళు ఉండొచ్చులేమ్మా, నువ్వెవరితో మాట్లాడతావు? అందరికి ఉండి నీకు ఇంట్రడ్యూస్ చేసినా చెప్పి చేసారు. మినిమం చేసి పెట్టాను. 140 ఇచ్చాను మరి 1కి బ్రేక్ ఇచ్చే వాళ్ళు 1కి 2కి రావాలి కదా 145 వచ్చేయాలి కూర్చో ఇంట్రడ్యూస్ చేసి చేస్తాను ఇదిగో మినిమైజ్ చేసి పెట్టాను కాదా బ్రోషరు నీ బయోస్కెచ్ చదవాలి మామూలుగా చెప్పు ఇది ఎంత ఉందేంటి 2 నిమిషాలు అంతే ఉంది నాకు చదవడం నిమిషం పడుతుంది delegates and dear students, now we'll be proceeding on with the next lecture. So for this we have our Dr. C.H. Shanti Devi. She'll be talking on the recent advances and future perspectives in environmental microbial biotechnology. Dr. C.H. Shanti Devi is a regular lecturer and head in the Department of Microbiology, Common Degree College, Men, Srikakulam. She did her master's degree in Kakatiya University, Warangal. She has qualified for SLET in 2000 and completed MPhil in Botany from Andhra University in 2010. She has obtained her PhD in 2020 from Andhra University and published more than 15 research publications and 4 book chapters. She has organized several national workshops, seminars and conferences in the capacity of the coordinator. Her research interests are pharmaceutical microbiology, environmental microbiology, food and nutrition quality. So I request Dr. Shanti Devi Madam to please go ahead with her lecture.
Good afternoon, dear participants. Today my topic is recent advances and future perspectives in environmental microbial biotechnology. And coming to the environment, every organism in the earth is surrounded by a lot of things, said other organisms like microorganisms and plants, water, air, light, land, etc. These surroundings of the organisms, we will call it as environment. Environment consists of living and non-living things. Coming to the definition of environment, it is a combination of various physical and biological elements that affect the life of an organisms. The environmental word is derived from the French word environment, surrounding or encircle. The environment is the surrounding of all the living creatures. Environment to sum total of all and non-living things around the influencing one and another. Environmental science is the combination of the environment. It has both biotic and abiotic components and their interrelationship between them. Study of interrelations of organisms with the physical and biotic and abiotic environmental biology or ecology, we will call it as. Coming to the components of the environment, it consists of 3 components in the environment: physical, biological and cultural. In physical components, it has 3 components again: atmosphere, hydrosphere and lithosphere. Coming to the atmosphere, it is the gases surrounded to the earth. The gases surrounded to the earth is known as the atmosphere. Coming to the hydrosphere, the water content on the earth is known as the hydrosphere. Coming to the lithosphere, it is a rigid soil covering the environment. Coming to the biological components, we are having two types of components, abiotic and biotic. Abiotic, there are components which are non-living things included in the abiotic components. Coming to the biotic components, we are having again three: producers, consumers and decomposers. Producers are those which produces the food, that is the plants examples, and coming to the consumers, all man and animals will come under the consumers. They will eat the producers. Coming to the decomposers, mainly decomposers included the microorganisms like fungi, bacteria, and they refresh the environment every time. They are also known as the scavengers of the earth. Coming to the cultural component, we are having again three components: society, economy and politics. They will play very important role in the environmental protection. Coming to the environmental biotechnology, it is the application of biotechnology for solving the environmental problems, both the environment or man-made ecosystem. We are having two types of pollutions like natural pollution and man-made pollutions. Man-made pollutions are playing very important role for the polluting to the environment. So we are having the biotechnological applications for solving the environmental problems both in the environment and man-made ecosystem. The most important topic at the threshold of the 21st century are the environmental and biotechnology. We are having biotechnology in the environmental sustainability. Environmental biotechnology can be defined as the management of the environmental issues with the advance in the biotechnology like RDNA technology and GMOs. We are having three key points for the approaches to the biotechnology to detect the pollution and any other environmental changes by means of biosensor and biomonitoring. Biosensors are those which the biological component plus chemical component, we will call it as biosensor. Example for this is the pregnancy test and COVID-19 test. Coming to the biomonitoring, the toxic elements are in the body. The toxic elements in the body we will call as biomonitoring. The best example for biomonitoring is amphibians. Amphibians are acting as the biomonitors to prevent the unfavorable environmental changes in the manufacturing process by substitution of traditional process, and to control the remediate the emission of pollutants in the environmental. These are the key points for the protection of environment. The main importance of environmental biotechnology, why we need the environmental biotechnology is, environmental biotechnology needed to, first one is the eliminate the hazardous waste produced by the other technologies. By the industries which we are producing the hazardous waste, we have to eliminate those hazardous waste. And second one is the distinction between the similar species and ensure the species are not at the risk of extinction. We have to protect the environmental. By protecting the environmental, we can protect the all species in the earth, and that to create the alternative energy sources like biofuel. Biofuel is nothing but we have to produce the fuel by using the algal masses or bacterial masses. We can produce the fuel alternate to the petroleum. Coming to the contaminants which we are releasing into the environment are the solvents. Solvents are nothing but they high heavy metals, or there are two types of solvents, organic solvents and inorganic solvents. Organic solvents will include the toluene and benzene rings, etc. Coming to the inorganic solvents, we are having the sulfur gases, nitrogen gases, etc. Coming to the pesticides, they are composed of the chemicals which can kill the pests in the agriculture. Herbicides are those which can kill the herbs. They are the chemical components which can kill the herbs in the agricultural lands. And coming to the fungicides and insecticides, both are the they have the killing nature. Against, they act against the fungi and insects. These all are made up of chemicals only, which are poisonous to the environment. Coming to the petrochemicals also play a very important role in the contamination of the environment. And explosives like CNT and heavy metals like mercury always play important role in the environmental contamination. Heavy metals like mercury play very important role in the contamination of water. Behind that, they will consumed by the fishes, and after consuming the fishes, we will consume that heavy metals. By the consuming the fishes, we will get the diseases like meningitis. For problem solving, we are applying the biotechnology in environment. For the biotechnology is nothing but we are using the improvement of the organisms and to consumption of all the heavy metals. The most important threshold back help. Living organisms to develop the products, process the generate less waste and preserve the natural non-renewable resources and energy as a result of the following. First one is the improved treatment of solid waste and wastewater management. We have to improve the solid waste management and wastewater management by the microorganisms, and we have to use or recycle the water again. And second one is the bioremediation. Bioremediation is nothing but cleaning up the contamination and phytoremediation. Bioremediation is the nothing but cleaning up the contamination and phytoremediation by the microorganisms. When we are treating the microorganisms, we will go for the less pollutants released by that or absorption of the pollutants by the organisms or plants. In industries, they are preparing the green belt zone to absorb the heavy metals. And third one is in the and clean the production manufacturing with the less pollutants and less materials. We have to produce the environmental protective products like avoid plastics and avoid detergents, soaps, etc. Then less pollutant and less materials towards the environmental production. And production of biomass for energy production. For the energy production, we can use the algal or bacterial biomass like spirulina or cyanobacteria species can be used for the production of energy. And we have to imply the genetically modified organisms in the environmental production and control. The new environmental challenges and new approaches continue to gain the more group in practice, harassing the process of engineering for improvement biodegradation. We have to improve the process of biodegradation in simple method by the using of the bacterial cultures or microbial cultures. We have to degrade the pollutants and in a genetically engineering modified organisms. And reuse treated as wastewater. We have to reuse of treated wastewater after the sewage treatment. We have to reuse the water for the plants or for the purpose of all plants. And we have to use the biomembrane reactor technology. We have to use the biomembrane reactor technology. Thank you for listening this class. Any doubts you can ask.
Randamma, come in. మీరు ముందు ఇక్కడ జాయిన్ అయ్యారా? ఏది జాయిన్ అవ్వ లేదు మీరు. మన వాళ్ళందరికీ జాయిన్ చేయించండి. ఎక్కడున్నారు మన వాళ్ళు? కింద పొద్దున్న బయట ముప్పర్ మీటింగ్ పెట్టారు బాయ్స్ అందరు. నేను చూసాను, మీరు లేరులేండి అందరూ. ఆ గ్యాంగ్ అంతా పెట్టింది, మీకు ఇంకో గ్యాంగ్ ఉంటాయి కదా, గ్యాంగ్ 1 గ్యాంగ్ 2 గ్యాంగ్ 2 అంతా అక్కడ పెట్టింది. గ్యాంగ్ అంతా జాయిన్ అవ్వాలి, అందరితో జాయిన్ చేయించండి. ఎక్కడ అవుతున్నారు ఇప్పుడు? కింద ఉన్నారా మన వాళ్ళు? కింద వెళ్ళి అందరినీ జాయిన్ అవ్వమని చెప్పండి అర్జెంట్. ఏదో ఒకటి అర్జెంట్ జాయిన్ చేయించండి. Sandeepta ma'am, shall we start? I don't want to keep you waiting. I can join in the students as and when they come, ma'am. If it's okay with you, we'll start off.
Yes, sure.
Yeah. Okay.
Sure, we can start.
Yes, ma'am, because you've already come and it's been already late and I don't want to keep you waiting. So, good afternoon, dear. Yes,
No
ma'am.
problem.
Sure. Thank you, ma'am. Good afternoon, dear students. We have come to the afternoon
Can you
session.
hear me?
Yes, ma'am. We can hear you, ma'am.
Can you hear me?
Yes, ma'am. You're clear, ma'am. We can hear you, ma'am.
Hello.
Ma'am, Sandeepta, ma'am, we can hear you, ma'am.
Yes. Can you?
We can hear you.
You can hear? Okay. Okay.
Yes, ma'am. So, good afternoon, dear students.
we can start.
Yes, ma'am. Yeah. We'll be starting the afternoon
Okay,
session.
yeah, we can start, I said.
Yes, ma'am.
Looks
Sure.
like there is some time. Yeah, okay.
I think some problem, network connection, like I had already told you.
Yeah.
Yes, ma'am. So, right. Good afternoon, students. We are now starting the afternoon session of this popular DBT sponsored popular lecture series. And now we have the session for us, Dr. Sandeepta Burgula ma'am, Assistant Professor from the Department of Microbiology, Usmania University, will be delivering the lecture for this session. And before we start, I would like to give a brief bio sketch of ma'am. So, Dr. Sandeepta Burgula ma'am is an Associate Professor in the Department of Microbiology at the University of Usmania University, Hyderabad. She has had her master's degree in microbiology from Usmania University, a PhD in biochemistry from Usmania University. She has successfully handled 7 research projects from various reputed institutes including SERBS, TST PERS, UGC and DRDO. Ma'am has published more than 20 research articles and 3 books and presented more than presented several international and papers in international, national conferences and workshops. Madam has filed an Indian patent published on 15th October 2021 on gold nanoparticles for testing sepsis. She has been awarded with the Indo-US Visiting Research Professorship by American Society for Microbiology and Indo-US Science and Technology Forum to work at St. Jude Research Hospital at Memphis, USA. Madam has been admitted as an Associate Fellow of Telangana Academy of Sciences in July 2017. She has procured the Raman Fellowship for Postdoctoral Research in US for the year 2015 and 16. And she has been awarded the Certificate of Excellence, Honorable Jury Mention Research, 5th Academy Branding Awards, Educational Expo 2020 and the International Travid Award from DBT and ICMR for paper presentation at Hypoxia Molecular Memory of Oxygen, Sensing and Response Pathways held in Colorado, USA during 2010. So this was a very concise biosketch that we have given of ma'am. Ma'am, I think our students are very inspired to be in your presence, and I request you to go ahead with your presentation, ma'am. They have been eagerly waiting for you since morning. Ma'am, Sandeepta ma'am, can you hear me, ma'am? Ma'am, Sandeepta ma'am.
Thank you so much for your wonderful introduction. Yeah, I think it's a very nice opportunity to talk, especially with the BSc. Yeah, can you? Yeah, I can hear you.
Okay.
I'm talking. Can you hear me?
Yes, ma'am, we can hear
Can
you.
you hear me or not?
We can hear you, ma'am.
Yeah, I'm here. I am talking. Can you hear me?
Yes, ma'am, we can hear you. There seems to be a delay in my relay, but yes, we can hear you, ma'am.
Okay. I request Rajesh to please mute your audio because I can hear you talking. It's a bit distracting from here. Okay, thank you. So it's a nice opportunity to talk to you all today. And today's topic, as you might have seen in the brochure, is about emerging nanotechnology applications and in disease and its diagnosis and treatment. Okay, so I think since you are in BSc, maybe this topic might be a little new for you. So let's start with some basics of what is nanotechnology. Okay, so the first ever concept which was given by about nanotechnology was by Dr. Richard Feynman. He's a very famous physicist who also won a Nobel Prize, and he was the one who was talking about very small sized particles. And he made this famous statement called "there's plenty of room at the bottom," meaning that the smaller the size, the more space there is for any molecule. And the term, basically the term called nanotechnology, was given by Norio Taniguchi in 1974. And from then there have been several inventions and discoveries which have taken nanotechnology way forward. And in future we are hoping that this part of biology, which is quite disciplinary in nature, will be entering all parts of our life. Okay, so first of all, we are saying what is nanotechnology. Okay, although we are saying that Taguchi and Richard Feynman gave the term, basically they were referring to the size of the range 1 to 100 nanometers. And nanotechnology basically involves molecules and particles which are of this size in terms of several areas of biology and even other disciplines. Today we are mainly focusing on nanobiotechnology and in particular nanomedicine. So I think you know that biotechnology is a biological process or organism or system to manufacture products which are intended for our life, which will help us improve our life. Okay, and nanobiotechnology is also a part of biotechnology. It uses this nanobiotechnology uses nanotechnology to be applied in biological systems. Okay, and this system may involve usage of biological materials and structures to produce some functions. Okay, what functions we will see as we go on. So the nanomaterials basically is what is involved in nanotechnology. Okay, so nanomaterials are simply, again, as I said, they are going to occupy a space of less than 100 nanometers. And they can be of several types. Okay, they can be in the bottom of the screen. You can see there can be like carbon nanotubes, very thin and very long tubes. Okay, like gold nanoparticles. Gold, which is present as a salt, it can be converted into nanoparticles using chemicals and even microorganisms. Or they can be liposomes, very small sized lipid vesicles, which can take up some molecule inside them and carry them to the target site. Or they can also be quantum dots. These are like mostly gold particles are used for quantum dots. So these quantum dots can be coated with some material which will help in diagnosis or treatment. As we go on, you can see the examples. Okay, so the nanomaterials, as we have discussed, are of different shapes and in a range of sizes, and they can occupy the length or a scale in their dimension. Means very small spherical nanoparticles may be there, right? They are zero dimension nanoparticles because all the three dimensions are very small in the nanoscale range, which is between 1 to 100 nanometers. Or they can be 1D, where one dimension is in nanoscale and other two in macro scale, means slightly longer in one dimension, and in the other they are very small. Like you can see very thin nanotubes in the bottom, and nanofibers and nanorods. They are very, very thin, like maybe 1 to 5 nanometers in thickness, but they're much longer. So that's why they are called as one dimension nanomaterial. Then two dimension nanomaterial, you can see a very thin film. So just imagine aluminum foil, which is even more thinner and has a thickness of nanometers. And the other scale, that is length and breadth, maybe in macro scale range, but their thickness is very, very small. So they are in the nanoscale range. So they are called as two-dimensional nanomaterials. And the last one, no dimensions in the three-dimensional nanomaterial. There are no dimension at the nanoscale. They are quite big because they are complex structures. They are made of smaller nanostructures to form a one large structure like a dendrimer. Okay, so these are the classification of nanomaterials according to their sizes. Okay, so as I said, the nanoparticle is in the range of 1 to 100 nanometers, and compared to other material like proteins and human hair, you can see where they are comparing from this slide. You can see that it's 1 billionth of a meter. Okay, whereas the hydrogen atom is much smaller than 1 nanometer, whereas a protein is in the range of nanometer size. Okay, similarly computer chips are also in nanometer size, but human hair, which is looking like a carbon nanotube, actually is a very, very thick material compared to a nanomaterial. Okay, so like that there are several things about nanomaterials which are very important for us to understand, and because of their small size and shape, they can be exploited in terms of various properties. For example, they can help in enhancing catalytic efficiency of certain enzymes, how we will see. Then they have electrical properties, especially if they are made of metal, or they have increased electrical conductivity, especially if they are made of iron, for example. For example, magnetic nanocomposites, they are having iron in them. Okay, so that is one. Then they can have magnetic properties, they can have mechanical properties, they are very tough. Okay, they can have optical properties. For example, this property is very useful in producing fluorescence. I hope you all know what is fluorescence, right? So this is useful in diagnostics. Then they have spherical properties, like very hollow spheres, especially made of gold. Okay, they can be filled with other molecules for some things like drug delivery. Okay, or by using nanoparticles. Then they have some biological properties because for example, cancer cells, they are quite permeable to other materials which come inside them, unlike normal cells and tissues. Our normal cells are not much permeable, unlike biological, unlike cancer cells. So we can make nanoparticles which can be easily permeable to cancer cells and not normal cells, so that we can target killing the cancer cells and also for diagnosing the cancers. Okay, so today we are looking at basically the nanomedicine part of the nanotechnology. Although it has several applications, as you can see on the slide, like food and agricultural applications, then biotechnology applications, especially in uh, microbial biotechnology, then in engineering, robotics, in textile production, everywhere these are important. For example, we are having a collaborative project in the lab where we are trying to coat textile fabrics, PP material, uh, protective material for especially for COVID-19. People have been using PP, right? Polypropylene fabric, which is to prevent spread, be diagnosed or treated with any disease by the use of nanomedicine. Okay, so these molecular nanotechnology and molecular manufacturing protocols are very helpful in application of the field of nanomedicine. And why is this going to be used? It is going to be used simply for treating disease or diagnosing disease. Okay, for example, latest I hope you have all heard that there is something known as robotic surgery. Robots perform the surgery on the patient, not the doctor only controls the surgery through a computer, but the surgery is done by the robot, right? So that has helped in increasing the precision or accuracy of the surgery by minimizing other problems during surgery, like overbleeding or invasion of other tissues because of surgery. So all this can be prevented. Similarly, this nanomedicine will be useful in performing miniature surgeries, especially when the site of injury or repair is very very small and needs very high precision. Okay, for tissue reconstruction, for eliminating diseases, for example, cancer or heart problems, that is the blockage of blood vessels, pain, and so many things. So it will be very useful. And even though it is safe to say now that the area of nanomedicine is still beginning, there is a lot to be done in this area. And even now, there are a lot of things which have been done, and we will see one or two examples on the way. Okay. So basically, when we boil down nanomedicine to few areas where exactly it is going to be useful, I said surgeries like called medical robotics, therapy or treatment, drug delivery, which is highly specific, multifunctional therapeutics, for doing many things at the same time by diagnosing the disease and also treating the disease at the same time. If we use nanoparticles, it will be excellent, isn't it? Then imaging for diagnostics, then prevention and control of disease for by early diagnosis. Am I audible, Dr. Shanti?
Yes, ma'am, you're audible, ma'am. Ma'am, you're audible.
Hello.
Yes, ma'am, you're audible.
Okay. So I was wondering if I am clear or not. So I thought I'll just ask
Yes.
once. Okay. Okay. So let's see some applications or examples of applications of. Okay. Okay. So let's see some applications. So for example, okay, I'm talking. So let's see some molecular imaging using nanoprobes. Okay. So you can see in the picture on the side, right, how a mouse is there. A normal mouse is there in left side, and there is some mouse on the right hand side. You can see it has red, yellow, and green spot. It is basically reflecting a site of injury or site of tumor. Okay. So this is one technique of imaging. Using nanoparticles. So basically in this, what happens is we can quantify problems or physiological changes in vivo or inside the body of the animal or human being, okay, using imaging probes. These imaging probes are nanoparticles. Okay, they can be carbon nanotubes, or there can be hydrogels, and so many things. There are different types of nanoparticles. Okay, so these nanoprobes are basically coated by some fluorescent molecule, or they can be tagged with an antibody which binds specifically to a tumor. Okay, so by being specific, they help in basically studying the underlying mechanism of the disease, by understanding the progression of the disease, or by for understanding the response to treatment of the disease. Okay, so molecular imaging basically involves using nanoprobes which are labeled. So these labels could be, as I said, any fluorescent molecule, or here you can see an example, like iron oxide nanoparticles you can see here. When it binds, this iron nanoparticle is coated with the peptide which binds only to cancer cells. So once it binds the cancer cells, the magnetic property of the iron oxide enhances the images, okay, and helps in detection of that cancer cell. Okay, for example, by magnetic resonance imaging, MRI scan, it helps in enhancing the signal for an MRI scan. Okay, so that is one example of a diagnostic. So basically, nanotechnology in medical diagnostics helps with two things. Okay, first, for rapid testing. Second, to detect the disease at a very early stage, okay, than what current techniques can do. So definitely this diagnostics will be helpful, especially if they involve different types of nanoparticles according to what we require. For example, you can see here on the screen, there are silver nanorods which can be utilized, made of silver, gold nanoparticles, then carbon nanotubes and gold nanoparticles together, or magnetic nanoparticles. All these can be used. For example, you can see gold nanoparticles can be tagged with antibodies that particularly bind to a virus, like a flu virus. So it will help in diagnosing flu infection, okay, influenza basically. Or a silver nanorod, they can be used to separate viruses and bacteria. So you can make silver nanorods which specifically bind only to bacteria based on some molecule attached to them, and you can identify them in blood or any other tissue. Similarly, carbon nanotubes. Okay, carbon nanotubes along with gold, they can be used in detecting certain types of cancer, like oral cancer, by tagging them with some specific molecules which bind only to cancers, okay, specifically in oral cancer. Then magnetic nanoparticles, as I said, they can help in enhancing the MRI image of any tumor. So this has been studied in brain cancer and its diagnosis. Okay, so like that, there are several nanoparticles which can be utilized. So that is about diagnostics. Then coming to drug delivery. So nanoparticles can also be used to deliver drugs to the target site in the patient. Okay, so basically it is what this drug delivery is, a method for injecting or administering pharmaceutical compounds for treating disease conditions like Alzheimer's disease or cancer or heart problems and so many things. So here the drug delivery systems which are used are basically nanoparticles or nanomaterials, right? So these nanomaterials can be of different types. They can be dendrimers. Dendrimers, for example, they are basically complex structures. They're not simple nanoparticles, but they form a ball-like or a mesh-like structure by polymerizing. Okay, or they may be micelles made of any organic molecule. Or for example, bovine serum albumin, which is a biological nanoparticle. It has been exploited in making certain breast cancer drugs. Okay. Similarly, lipid-based nanoparticles. These are liposomes, are basically lipid vesicles, or neosomes, which are both lipid vesicles. They are basically small lipid vesicles which can be encapsulating certain bioactive components, like let's say antibiotic or cytotoxic drugs, which can bind only cancer. Or metal-based nanoparticles like gold, silver, cadmium, selenium, zinc, sulfur, etc. So in our lab, just to tell you, we are working on selenium nanoparticles. These selenium nanoparticles are trace elements. They are required in our diet in trace amounts. They are also highly antioxidant in nature. Okay. So, and also anti-cancer in nature. They have anti-cancer properties. So what we wanted to do in our lab, and we are to some extent we are successful in that, and we are trying to make selenium nanoparticles and coat them to generic anti-cancer drugs. Okay, and we found that at least in our laboratory, that selenium nanoparticles, when they are coated with anti-cancer drugs, are showing enhanced cytotoxic ability against cancers. So this is how drug delivery systems can be designed. Okay. For example, other examples you can see here: benefits of liposomes in drug delivery. As I said, liposomes are lipid vesicle. You can see the structure on the right side. This lipid bilayer is encapsulating some nucleic acid inside, isn't it? And also it is having some peptide coated on the surface, which will specifically bind to the target cell. Okay. And also this has a very small amount of drug encapsulated inside the lipid vesicle, which will kill the target site or target cell. Okay. So liposomes are basically spherical vesicles. As you can see, they have low toxicity because they are basically made of lipid. Okay, they're capable of displaying the drugs on the surface, as you can see, and also they can encapsulate the drugs. So examples are cationic liposomes. They have been utilized in non-viral gene delivery to condense DNA. Okay, so here I would like to show you a short video. Okay, so for example, I was talking about the drug. I think you can see my screen, Dr. Shanti. I'm trying to show a video.
Yes, ma'am. Yes.
Okay.
Scientists are using cells isolated from human tumors to develop new ways to tackle cancer. Here we can see the cancer cells growing in a culture dish in the laboratory. For most drugs to be active, they must go inside the cancer cell. Scientists are developing tiny particles, nanoparticles, that can be loaded with drugs. The nanoparticles can be modified so that they attach to the cancer cell and get carried inside. To make sure that the drug-loaded nanoparticles only attack the disease cells, they are engineered so that they only bind to the target cancer cells and not to neighboring healthy cells. Once they bind to the cancer cell, they are taken into the cell. They can't pass through the surface. Instead, part of the cell surface forms a pit, and then it engulfs the nanoparticle. Now we are looking inside the cell. Cellular proteins wrap around the vesicle and drive it inside the cell. You can now see a vesicle carrying the nanoparticle moving towards the very center of the cell. The vesicle carrying the nanoparticle travels further inside the cell and then fuses with a compartment called an endosome. This is part of the digestive system of the cell's stomach. The inside is acidic, and its job is to digest incoming material. The nanoparticles are degraded inside the endosome, releasing the drug into the cell. The drug can then kill the cancer cell. Nanoparticle-based drug delivery provides a way of attacking cancer without the side effects of conventional chemotherapy.
So I hope the video was audible and interesting to all of you. So it was such an attractive way to show that, the vesicles which you are seeing here, how they enter the target cancer cell, right? The peptide coated on the vesicle basically helps in binding to the cancer cell specifically. Okay, and since the lipid vesicle is made of lipid, obviously, it easily fuses with the cell membrane of the target cell, and it is internalized, and inside it goes into endosomes, and there it is digested, as you could see in the video. Once it is digested, the drug which is encapsulated is released, right? And this releases the toxic material against the cancer cell, and this drug causes the death of the cell. And since it is only going inside a cancer cell and not the normal cell, this way of drug delivery is supposed to be very safe and useful in treating cancer. Okay, so the advantages, to sum up from that technique, as we have seen previously, is that the drug is basically attracted to only diseased cell, which allows direct treatment of only diseased cells. And therefore, the application of drug delivery by nanotechnology will be helpful in increasing the level of drug delivery to specific cells. For example, tumor cells or any other damaged cells. They will help in sustained drug delivery using a hydrogel. A hydrogel is basically made of nanomaterial, which is difficult to digest. Therefore, the drug inside the hydrogel is released slowly and it is sustained. It does not stop for a long time. So that sustained drug release helps in reaching the target cell. Okay. And the nanoparticles, if they are made with by a complex method, they can have multiple, anti-cancer drugs encapsulated inside them or tagged to them. Okay, nanoparticles can also be used to deliver nitric oxide directly to cancer cells, which causes the death of the cancer cell. Nitric oxide is harmful for cancer cells, unlike normal cells. So it can be used here also. Then carbon nanofibers, as I had mentioned previously, are one-dimensional nanomaterial. They can penetrate cancer cells easily, and they can deliver anti-cancer drugs. Okay. For example, you have seen cancer previously. Now let's see an example of heart disease. So in heart disease also, what happens is there is a blockage of blood vessels like arteries. So parts of arteries are damaged and there is a blocked artery, in such a site. Especially many times, like in embolus or ischemia or blood clot, right? So for this, what people have done is they have made nanoparticles using iron oxide that allows nanoparticles to be directed by a magnetic field to stents. Stents are also made of some material, isn't it? So these iron oxide made nanoparticles have some magnetic field which will help them find stents. Okay, and these nanoparticles can be coating certain blood clot dissolving drugs, or enzymes. Okay, so that once they reach that home in, or that they reach inflamed tissue such as arterial plaques or blockages and dissolve the blood clots. Okay. So that is one. Then, for example, we are talking about diagnostics and therapy. Another way of doing therapy is to use the nano-sized particles responsive to certain fields like electromagnetic field or acoustic excitation, that is sound. Okay, ultrasound. Okay, so once they reach the target site, they can tolerate extreme condition like heating, but not the biological material nearby. So once they reach the target site, they are specifically heated so that the target site may be killed or damaged and the problem can be treated. Okay, some memory loss, then loss of muscle coordination, and so many other problems. Okay, so this amyloid beta is accumulating in brain, isn't it? So to target this, people have tried to use functionalized gold nanoparticles which selectively attach to the amyloid protein. Okay, you can use an antibody or any other molecule which has affinity to this protein. Okay, then they have functionalized the nanoparticles in a way that the microwaves of certain frequency are irradiated on the sample or the nanomaterial. Okay, so resonance with gold nanoparticles, what happens is it increases the load because the quantum dots are coated by certain material or antibody which goes and binds to the tumor cells, and they are also tagged with other material which will absorb infrared light, okay, to give images which can help in localizing the tumor. Okay, so this picture I had shown you previously, right? So these quantum dots can be, as I said, covalently linked to peptides or antibiotics or any other molecules like nucleic acids. They have more surface area, and therefore the intensity of the signal is quite high, and this signal can be captured. Okay, so different signals are made by using different fluorescent molecules or nucleic acids or antibody. Okay, so for example, here you can see there can be multiplex diagnostics by using different quantum dots, okay, of different diameter and different fluorescent properties, okay, to distinguish between two different phenotypes, particularly in cancer. Okay, for example, you can see here how aggressive cancer cells are showing orange fluorescence, whereas mild cancer cells are showing pinkish to red fluorescence. Okay, this is because of the tagging of the nanomaterial with the different molecules which will have affinity to different types of cancer cells. Okay, that is called as multiplex uh, mode of using nanomaterial. Okay, so like this, there is a very new development where people have been using, and this is the only nanoparticle based drug which has been patented and is used in the market for cancer. Okay, so women is going to bind to cancer cells and help the paclitaxel go inside and kill the target cell. It is helpful in targeting particularly breast cancer cells. Okay, so that is about anti-cancer therapeutic molecules using nanoparticles. Now let's look at something even new, which is called as nanorobotics. Nanorobotics are very interesting. I don't know if you have seen the new movie of James Bond. Has anyone seen new James Bond movie here?
Yes, ma'am.
Very good. So you have seen the movie. Have you seen how James Bond is injected with the nanobots,
Yes,
right?
yes, ma'am.
So those nanobots, they made using the person's using James Bond, genetic signatures. Okay, all of us have genetic signatures on our chromosomes. Okay, so what that villain does is he takes the genetic signature of James Bond and he tags it to a nanobot, which is toxic. Okay, so as soon as the nanobot enters James Bond's blood, okay, so the way the genetic signature is designed is that if he touches anyone who has similar genetic signature like him, if he touches them, these nanobots will activate and he will die or they will die, something like that, isn't it? So these are nanobots or nanorobotics. This is a future area of nanomedicine, but people are slowly exploring this area of nanotechnology. Okay, it is a technology of creating small machines of nanoscale size. Okay, and these are basically made up of carbon in the form of fullerene, which is very interesting carbon material, or diamond. Fullerene is easier to use. It can be made in the lab. Okay, and it's very strong like diamond, which is also carbon material. Okay, and this application of nanorobotics is that they can easily flow through human body because they are very very small. They can enter circulation. Okay, so such nanobots, if they're programmed properly, they can be used in doing miniature surgeries like I mentioned in the beginning. Okay, they can help in finding the danger cell or the toxic cell or the villain, okay, inside our body. Okay, so that once it goes and binds to the dangerous cell, it will release the toxic compound like you have seen in the video I have shown you, right? And it will kill the target cell basically. Okay, so it is like a spy. Okay, spy or a detective. It will go and find the villain and also kill the villain. Okay, isn't that interesting, right? If it is done inside our body without our own knowledge. Okay, and since it is very small, it can enter the cells. Okay, it can insert the correct copy of DNA inside the chromosome if it is defective. or if there is no gene present inside the chromosome for a particular disease, so it can replace it. Okay, it can help in miniature surgery or repair of tissues. It can break up blood clots, as you have seen previously in previous slides, or even dissolve kidney stones. It can help in treating cancer. It can also help in continuous monitoring of glucose, especially in diabetics, right? You know that one in five persons nowadays suffers with diabetes. So in such case, the treatment cost is increasing day by day. So if you can continuously monitor your own blood sugar levels, you don't have to go to the diagnostic center and wait for a day to see your readings, or even use AccuCheck, which is a portable glucose measuring device, right? You don't have to use it. Your body can be attached to a small chip which will continuously monitor glucose. Okay, so applications of nanorobotics, as I said, is in surgery, especially if the nanobots can be attached to a very small nano camera, so that the visualization of the surgery inside the body is easy. Okay, and since it's a nanobot and it can be precisely controlled from a computer, there will be very less chance of mistakes. Okay. Okay, so look at the idea of using a nanobot here. Okay, so this nanobot here is entering a lymphocyte, and it wants the lymphocyte to die because the lymphocyte is defective. It may be cancerous, okay, or it may be infected by HIV. HIV, it affects T lymphocytes, isn't it? It injects a molecule which is toxic to the lymphocyte and directly kills it. For example, another nanobot, what it does, it makes a small hole inside the small tumor. Okay, and it delivers its drug which is inside the nanobot. After drilling the hole, the drug enters the in the small tumor, thereby killing the cells inside the small tumor. So it prevents the growth of the tumor. Okay, so like I said, nanobot can also be helpful in taking blood sugar levels continuously, or it can find and kill bacteria directly inside the blood and prevent infection. Or you can see here, it is this virus finder nanobot is directly going and finding the virus and trying to kill it. Or like I said, nanobot can also be tagged to a blood clot dissolving uh, molecule. On the left hand side you can see a nanobot is going to an atherosclerotic deposit in the blood vessel, and it is dissolving that atherosclerotic spot or plaque. Okay, and similarly, dentists are also trying to use nanoparticles or nanobots or nanorobots to clean the surface area of patients' teeth, especially near the gum line, to prevent tartar or any cavities. These can also be explored in future. And this example is very interesting, as you can see on the slide. Which is called a respirocyte, right? It's a proposed nanorobot. People are trying to make this. Basically, they are trying to make an artificial
no
red
phones
blood
in the
vessel,
class.
the red blood cell. And what this red blood cell will do is it can have oxygen binding capacity, which is more than 200 times than RBC. Okay, it is much higher. So people who are suffering with, let's say, COVID, they are very short of oxygen. So such people can be injected with this respirocyte, so that this respirocyte can transport much more oxygen. So without the use of oxygen cylinder, it can help increasing the oxygen levels, dissolved oxygen level in the patient. Okay, so this is useful also in the treatment of certain genetic disorders like sickle cell anemia. Then in blood transfusions, it may be helpful in maintaining the blood oxygen levels, and so many ways in fetal oxygen related disorders, wherever hypoxia or low oxygen levels are causing a problem. These respirocytes can be used. They are very, very small. They're hardly 1 micrometer in size, so they can be injected into circulation. Okay, and they can be utilized. Similarly, lab on chip, new technology like nanobots, there is something known as a lab on chip, which is a miniaturized diagnostic device. These give very fast diagnosis using very small amount of material or fluid. Okay, and the lab on chip, or these devices, they'll be very useful because the samples which have to be analyzed need not be sent away to any lab, but simply these chips can be utilized directly at point of care or with the doctor to do any diagnosis. Like for example, chips like microarrays or biosensors, they are helpful in detecting amount of any defective gene or defective copies of genes or proteins or any molecule inside the sample which is obtained from, let's say, a diseased patient. Okay, so in our lab, in this context, we are trying to make, we have recently patented one test using gold nanoparticles. It is used to diagnose sepsis. Sepsis is caused when bacteria enter blood and they release their endotoxin, which causes massive hemorrhage of internal organs. Okay, it leads to death sometimes very rapidly. Within a few hours, a patient can die. So nowadays, the diagnosis of sepsis takes half a day to one whole day. Sometimes the blood cultures take 2 days to diagnose any infection. So in that time, many cases, what happens is the patient can die. So we have made a test in our lab which will detect sepsis within minutes, and it is 1 or 0.01 ml of serum obtained from the blood, and this helps in diagnosing the sepsis within like 10 minutes. So we don't use any major equipment, and within few minutes we can diagnose. And we have just patented this test, and we have used gold nanoparticles to devise the test. So this will be useful in diagnosing sepsis rapidly and with low cost. Okay, so like this, there are several lab on chips, lab on a chip assays which can be performed using nanoparticles. For example, in case of biochemical assays, immunoassays, we can design using lab on a chip technology. Then chemical applications by separating molecules from mixtures can be done by this method. Okay, by biological application, that is co-culturing different types of cells together to study a system, let's say a cancer or a tumor, and so many things we can study. Okay, so I know that 1 hour is not sufficient to understand the in-depth applications of nanoparticles, but basically what I want you to understand is that these nanoparticles can be utilized for both diagnosis or treatment, right? But in future they can also be used for making scratch resistant paints. So your cars, which are easily scratched by, if you write with a pen or anything, you will get a scratch on the car. But nanomaterials can be made which are resistant to scratches. Now people are trying to use it in very high end cars. Okay, to make very efficient fuel cells and batteries also people are trying to use. Okay, and this is lot of money. Actually, when we are talking about nanotechnology, there is lot of money, and future will decide how much money or how much revenue this technology will generate, because especially in US alone, in next 10 years, people are expecting that at least 3 trillion US dollars will be invested in this area of technology. Okay, so as everything has its positives, this area of nanomedicine particularly has also some drawbacks. Okay, because as I said, they're made of metals or some nanotubes. They may have some toxicity against our body, so they may cause some serious problems, especially if nanoparticles like gold or selenium start accumulating in our body. They can cause damage to our human body. And in some cases, like I was telling you about the James Bond movie, if these nanobots are so small, that villain makes them in a way that you cannot even find them easily in the blood. So they are untraceable, and we don't know if he wants, he can inject into anyone and decide to kill anyone. So it's a destructive weapon to use such, nanoparticles. Okay, and since this is an expensive technology, probably in at least in this stage and even coming future, only rich people can afford it. So there is a very big gap in terms of treatment of patient, because only if only rich can afford, then the usage of this technology will be limited. Therefore, Still a lot of time is required for studying these. They also cause a political dilemma, so people may, if especially people at higher level will wonder if we should allow implementation of such expensive treatment methods because they are only useful for certain class of the society. So these are some implications. Definitely in future, these will be overcome, and uh, much more number of people will be definitely benefited with the applications of nanomedicine. Okay, so finally, what we would like to conclude is that this nanotechnology has very big scope in building large number of products that are useful in diagnosis and also treatment, which can be quite powerful, especially looking at the way you can see how drugs can be targeted towards the site of tumors or damaged cells. This has a very big scope. Okay, and this area is heavily interdisciplinary in nature. Okay, therefore its scope is quite large, especially in cancer. It has a very good scope because specifically the problem today is that anti-cancer drugs can kill all cells, including healthy cells. But if you could target only cancer cells, there is a very good chance that the side effects of anti-cancer drugs will be reduced drastically and also improve the healthcare against cancer to a much better level. Okay, and also the diagnostic tools, they can be improved and they can be utilized using nanotechnology. And of course, as I said before, there's still a lot of scope for improvement, and also the implications which are there for nanotechnology, they have to be addressed in future to give a better scope of this area. So this is about my talk today. This is my lab. I hope you can see the photograph. These are my students. So the boy at the back is Karthik. He's working on selenium nanoparticles. He recently published in a very good journal on selenium nanoparticles and their anti-cancer activity. And the girl on the left side, who's famous, she's Sandhya. She also worked with gold nanoparticles, and she's the one who has helped make the nanoparticle-based diagnostic test for sepsis. And the boy on the right side, Ramesh, who's wearing a checked shirt, he's continuing this work on sepsis diagnostics, and he's trying to test our diagnostic test which we have made on more number of patients. So this is about my talk today. I hope my talk was clear.
Yes, ma'am. It was clear. And I think you were very clear, in fact, in the sense that we are lacking any queries from our students. So thank you, ma'am. Thank you, Sandeepta, ma'am, for your very illustrative lecture. Clear. The concept was very clear in the video, ma'am. I enjoyed the video. And you also simplified the entire concept of nanomedicine, including the
I hope.
Yes, ma'am. Ma'am, there seems to be some problem during from our side. My voice seems to, come across to you very late.
I hope my voice was clear because I think there is some lag in the audio. I hope everyone could understand the video also, because I thought, yeah. Even mine is coming a bit late. But I hope my voice was clear while presentation at least, and all of you could see the video and all clearly. If you have any doubts, you can always contact me on my email ID also. Okay, or you can talk to me on my WhatsApp number. I think Dr. Shanti has it. So that is about nanotechnology.
Yes, ma'am. So thank you, ma'am. I'm sure most of our students were inspired through your innovative ideas of how this nanotechnology can be applied in diagnostics and in treatment too. So thank you, ma'am. It was very nice of you, and you had waited in spite of all these technical difficulties that we are facing today. You bore with us, and you were there for our students and for us. Thank you, ma'am. I'm Dr. Harita from Biotechnology Department. Shanti, ma'am, actually is not feeling well.
Sorry,
today.
I couldn't hear you. Sorry, I could not hear you.
Yes, ma'am. Ma'am, I'm Dr. Harita from Biotechnology Department.
Oh, okay, okay.
Yeah.
Okay.
Yeah. Shanti
Okay,
ma'am
thank you.
actually wasn't feeling well.
So yeah, so if you have any other questions, you can always contact me. And yeah, so they're all
Simplified. In fact, the medicine, nanotechnology itself is a very complex topic, and thank you for joining us. Right, dear students, with this we come to the end of the popular lecture series sponsored by DBT, and I would like to propose a formal vote of
okay.
thanks.
Bye.
At the very onset, I would express our deepest gratitude to the for her encouragement and her support for the program. And